Okay, so I'm going to continue with the theme we started last time, which is how chromosomes behave during meiosis. And we'll start just by reviewing some of the genetic aspects of meiosis that we talked about last week. That's what we did, isn't it? Can you see that okay? Is that right? Okay. I'll just try to, you don't have to read everything about it. We talked about how people figured out that inheritance was mediated by chromosomes, and that chromosomes contain genetic material. And the main piece of observational evidence for that, at least at the beginning, was that chromosomes obeyed the same kind of doubling in number and reducing by a factor of 2 that Mendel's genetic factors also did. That's what led people to suspect that chromosomes were really the genetic material. They did a lot more experiments too to confirm that. In more detail, the fact that 2 chromosomes from the mother, one from the mother and one from the father, that might have different genes on them, they segregate to 2 daughter cells, is the same thing that Mendel saw when he knew that there was, for instance, a yellow color that might have a hidden green color gene inside, and those would go to both daughter cells. And the fact that genes can associate independently, so the color of a plant will assort or travel independently of the texture of the seed. The fact that characters can segregate independently is directly reflected in the fact that there are more than one chromosome. There's more than one place for genes to be located on, and different chromosomes do not interfere with the segregation of other chromosomes for the most part. And then later it was learned that not only do chromosomes contain genes in them somehow, but that genes occupy very specific reproducible positions in a line, so that one gene is followed by another gene, followed by another gene. And then also there was this idea of dominance and recession. So we have different phenotypes give rise to different genotypes, but sometimes there's a hidden genotype. So this small r that will specify the texture or the skin of this seed does not, it only shows up when both alleles of this gene are small r. And so it's, in common English usage, people sometimes talk about dominant genes and recessive genes. So they may say, or dominant alleles and recessive alleles. And I think this word allele is maybe one I haven't really emphasized. So does anybody know what allele means?
No.
No? Okay. Do you know what the word allele means? No, not quite. Yeah, it's related to homologous chromosomes. So allele is just a kind of a fancy word for the type of gene. So big Y and small y are 2 different alleles of the Y gene. So it's kind of like the kind of gene you have.
And is it 対立遺伝子?
対立遺伝子. Yes, yeah. So you have homologous chromosomes, 相同染色体. So there's one from the father and one from the mother. And then this would be 対立遺伝子, right? So you have yellow on the father, and then maybe it's a different version of yellow on the mother. So these are different versions of genes. So when we have 2 different versions of genes at the same location, the very same location on the chromosome, these are called alleles. So a gene can be said to have many alleles, and genes can come in more than 2 alleles. Right? There can be many different versions. This might give you very bright yellow. This might give you no yellow at all, so you turn green. But there might be another kind of allele here, which is Y prime, which gives you a little bit of yellow, but not much. So it might be an intermediate yellow. So a gene can exist in many different versions or kinds. And so those, when we talk about the kinds of genes you can have at the same position, those would be referred to as alleles. And that's different from, for instance, you might have another location on a different chromosome, on the same or different chromosome that also controls the color. And maybe the genes here also affect the yellow color of the seed. You can have multiple genes affecting the color. But this would not be an allele of this Y gene. This would be a different gene altogether because it's not at the same position. So, so allele is you have different kinds or different versions of the same gene at the same position. And so it's easy. It's a little bit. It's a very common thing for people to say, okay, the capital Y is a dominant allele or dominant version of this gene, and the small y is a recessive allele. But the problem is that genes can sometimes do more than one thing. So this gene may be encoding a yellow pigment, and it may be dominant in the seed, but it may, for instance, it may turn out to be also affect the color of the leaf, and there the dominance and recessiveness might go the other way, because who knows what other genes are interacting with this gene in the leaf versus the seed. So the environment that this gene acts on plays a big role in whether it's dominant and recessive. So when we talk about dominant and recessive, we are really talking about the phenotypes. Okay, so we can say that yellow seed color is dominant to green seed color with respect to the Y gene. So the Y gene itself is not dominant, but the phenotype, so this phenotype is hyogengata, I guess. It's the expression of the gene in a certain context. Those can be dominant or recessive. So it's a little bit complicated to say. It's not as easy as saying yellow, the big Y allele of the yellow gene is dominant. But we can say that yellow seed color is dominant with respect to the Y gene. That's much more complicated to say. So sometimes we say that capital Y is dominant if we understand what we really mean. Okay. And likewise with recessive, the small r, for instance, for the wrinkliness is not necessarily recessive because in another context, maybe the wrinkliness of the leaf, maybe it's dominant. Okay, so it's just important to keep in mind that dominant and recessive are not properties of genes or alleles themselves, but they're properties of how this gene is interacting with the environment that they're expressed in. And in this case, we're talking about the environment is the seed, the outer part of the seed here. And then the next bit we talked about was that since genes occupy given specific locations on chromosomes, when you inherit those, when you put those chromosomes into the next generation, the chromosomes can choose which alleles go into the next generation, and the possibilities are the parental combination. So all the alleles that are on one parental chromosome can go into the next generation, or you can also have crossing over between these alleles. So you can start the DNA molecule here on the left, and you can have a crossover somewhere in here that continues that way, and then you have capital Y allele, and then the small r and the small p allele. So you can get these kinds of alleles with a single crossover, and then you can actually, in some cases, cross over once and then cross over twice to get a combination involving two parental and one non-parental allele in between them. And the fact that these crossovers happen at different frequencies was a direct reflection of the fact that genes occupy different positions and there's different distances in between these genes on the chromosomes. So genes are very close together, then it's very unlikely for a crossover to occur here. Whereas in this allele, which is in this interval, which is 3 times bigger than this interval, you have crossovers 3 times more likely to occur, which means you would be more likely to have this combination than that combination. You're more likely to keep these 2 parental alleles together and cross over here than you are to keep these 2 parental alleles together. Okay. So that's those are some of the things that we went over in genetics. And later I'll be talking a lot about crossing over, but not actually showing what's happening. I'm just saying somehow these alleles go together, and then you somehow pick up this allele. But I haven't shown you what is actually happening to the DNA molecules in order for that to happen. But we'll go over that in a later class when we talk about the molecular determinants of how crossovers happen. For now, we're going back to some basic aspects of chromosome behavior during meiosis. Again, the things that chromosomes have to do in meiotic prophase, in the prophase part of the meiotic cell cycle, all has to do with getting these chromosomes together so that they can pull away from each other and be segregated into daughter cells. So in prophase, these various things have to happen. Chromosomes first have to find each other. They can start out completely separated from each other on opposite sides of the nucleus, but they need to move to find each other. Once they find each other, they need to pair. They need to know, okay, you're my correct partner, and I'm rejecting all the other partners. After they've paired, then they need to synapse, which means they come together really, really tightly, and they get a protein zipper between them, so that all the homologous DNA is next to each other. Then they do this recombination. That's still a kind of mysterious black box that we'll talk more about next time. But this is how the crossing over occurs. And the crossover is required in order to get successful chromosome segregation. So if the chromosomes do not cross over, then there's nothing holding them together, and they'll just fall apart again. So recombination not only creates genetic diversity and shuffles the alleles of genes, but it's also required to hold chromosomes together. Okay, so I've shown you a lot of diagrams of chromosomes, but of course they don't really look like that. I've shown you some real images of chromosomes, but the next thing I want to show you is the different stages chromosomes go through in meiotic prophase. And to show you that, it's best to show you an organism with very large chromosomes. And this is a plant. I think it's a lily. I have to check to make sure, but I think it's a lily or some other kind of ornamental flower with 12 chromosomes. The first thing to show you is that these are all nuclei of the same species, just different stages of meiosis. So the exact same amount of DNA is shown in all these pictures. It's just the DNA is undergoing different types of organization on its way to the meiotic divisions. So A should remind you maybe of the electron micrographs of euchromatin and heterochromatin that I showed you, and that's because A is simply an interphase cell with no real chromosome condensation going. So all the chromatin is just kind of not organized in any loops yet. And it's just to recall, does anybody remember what this dark area is called? Yeah.
The nucleolus?
Yeah, so this is the nucleolus, and so why is it dark? So
No DNA.
yeah, that's right. So there's no DNA in it, except you can see this little bit of DNA here, and this is probably the little bit of DNA that is involved in transcribing all this ribosomal RNA that makes up the nucleolus. Okay, so you can see then, I hope you can see there's a difference between A and B. In B, you start to get these little tiny threads of chromatin forming. And so B is when meiotic prophase really is starting. So prophase is when chromosomes start to condense in preparation for cell division. In mitotic cells, they kind of go directly from this to something that looks like this. But in meiosis, because chromosomes have to line up next to each other in preparation for recombination, they don't just condense down into little rods, but they remain as very long threads, and that's what you can see happening in B. So B is a stage called leptotene. I'm not going to expect you to remember all these stages, but I just want to tell you that meiotic prophase has been broken down into various sub-stages that are named based on the appearance of chromosomes. So just by looking at chromosomes, there's some very drastic differences that happen at different times, and they go in this order in time. So they start with this stage called leptotene, and you can see they all have this word "teen" in it, and I think I mentioned before, this just means thread, because when people first looked at it, they just thought they were seeing these threads of material. So leptotene is very light threads. That's what that means. So, and that's true. So this is like the lightest, thinnest threads that you can see. C is the next stage. It's pretty drastic going from B to C. C is the stage called zygotene, and zygotene means joined threads. So threads that are joined together, and that's because chromosomes are starting to pair. So then after zygotene, another pretty drastic change happens, and that's D, which is, you can see that there's the exact same amount of DNA, it's a DNA. The exact same amount of DNA between D and B. It's just that it's become more condensed in D. Instead of thin threads, we have thick threads, and that's what pachytene means. So the threads have become thick. This is the stage when chromosome pairing and synapsis are completed, so there's no more searching going on. All the chromosomes have found their partner, and this is the stage when recombination and crossing over happens. So we'll go on a little bit to show some diagrams of what's happening. So these are individual nuclei now of C. elegans, where this is kind of a diagram of what's happening. So interphase chromatin looks like this. So before any condensation is happening, the chromatin is just kind of randomly going throughout the nucleus. But these lateral elements act, so there are these proteins called lateral elements. Among them is condensin and cohesin, these proteins that act during to hold sister chromatids together. And these start organizing the chromosomes into these loop structures. And it happens kind of randomly all along the chromosome length. And that process continues. So these short stretches of condensed chromosomes then kind of build up into longer and longer stretches. And so they get these kind of long threads. So this is how leptotene threads are made. There's another protein that is involved in holding chromosomes together, and that's, I guess I should go to the very end. I should skip to what that looks like at the very end. So there's this protein complex called the synaptonemal complex that once chromosomes have paired and come together, so this is, say, the chromosome from the father, and this is from the mother. Once these chromosomes are together, there's a protein zipper that goes down the middle, and it actually zips up the chromosomes. And if you watch this happening in living cells, it really starts at one end and starts to zip up the two chromosomes together. So that's what I'm showing in this one right here. Okay, so the synaptonemal complex starts at one end and starts zipping these chromosomes together, and that's why you go from these thin threads to these thicker threads, because each thread now consists of two chromosomes that are tied together. This process finally goes all the way from one end to another end, and all the DNA is completely packed together. And with microscopy, you can see what this is in structure. So these lateral elements that were making chromosomes condense are held together by these central elements that really act as this kind of train track structure that just bridge the lateral elements. And you can see with microscopy, here's the green lateral element and the central element right in between them. And the blue here is the DNA that's coming out from that train track in these loops. Okay. And I won't really get into it, but this SC, the synaptonemal complex, this structure, is actually composed of many, many proteins. There isn't just one central element zipper protein, but there are many proteins. In C. elegans, there's 4 of them called CYP1, CYP2, CYP3, and CYP4. Different organisms have different assemblies of this central element. It seems like it can change a lot, but all they really have to do, there's a really just structural building block proteins. All they really need to do is just sit there in the middle and hold these 2 axes together. All right. So these are the things that have to happen during meiotic prophase. And so the last 2 stages, I'll probably get to later because they involve crossovers. But after this Pachytene stage, there are 2 more stages left called diplotin and dikinesis. And those are stages that happen after crossing over has finished. So recombination finishes, and then once recombination is done, there's no need for this synaptonemal complex anymore. So it gets removed, and then chromosomes are free to condense all the way down. So this stage is the next stage, diplotin, and you can see chromosomes get really, really short. So the synaptonemal complex is kind of keeping these chromosomes very long, and once it goes away, chromosomes just kind of condense down, and you can see these. Here's a good example. You can see that there are 2 crossovers. So there's 2 points at which these chromosomes are touching each other, and that's where they're exchanging DNA segments. So diplotin and dikinesis are the stages where you can see those crossovers very nicely. Okay. All right. So anyway, I just wanted to give you an idea of how much chromosomes change as meiosis proceeds. And I showed you some of the proteins involved in the changes, but actually there's really a lot more going on. And the transitions from here to here to here are not, they don't only involve chromosomes just loading onto, proteins just coming onto chromosomes and changing them, but there's also a lot of feedback, so the chromosomes check to make sure that this step has happened correctly before they go on to the next step. So there are a lot of layers of regulation. Chromosomes don't just collapse, because if they did, they wouldn't make these really nicely resolved threads. So the process of condensation is very highly regulated. Okay, but it's nice to show you these really big chromosomes to give you an idea of the drastic changes that chromosomes undergo during meiosis. But for biological study, really a smaller organism is better. And I showed you last week that I'm studying C. elegans, this little nematode worm, because it's really small and you can see its chromosomes very well. There's certain special things about meiosis in C. elegans. In fact, every organism has some special aspects of meiosis, including humans. But some things that are universal are, just like every other organism, C. elegans chromosomes have to undergo pairing and synapsis and recombination and crossing over. So all the really important aspects of meiosis are present in C. elegans. So by studying them in this tiny worm, we can learn about how those same processes work in larger organisms like people. However, there are several non-standard aspects that exist in C. elegans that are different from people and from other animals. And I'll go over one of them at the beginning. So let's see. So in most organisms, including us, chromosomes need to move together in order to encounter their homologous partners. So let's say this chromosome and this chromosome need to meet up with each other, but they start out separate from each other. So what happens in most organisms is the telomeres, the ends of the chromosomes, like I said last time, they attach to the nuclear envelope and they undergo this motion. And so the chromosome ends all come together in more or less the same part of the nucleus. Once they've come together, they can recognize each other and then they recombine and they can segregate the genome in meiotic divisions. And humans, for instance, let's say this is human chromosome 1. You can look at, maybe this would be the right end of human chromosome 1, and this would be the left end of human chromosome 1. And both ends are attached to the nuclear envelope, and both ends will undergo this motion to find each other. But in C. elegans, they do not do that. They do not use the telomeres. They don't use the very ends of chromosomes to attach to the nuclear envelope. But instead, they use something kind of similar that's called pairing centers, which is not the very end of the chromosome, but it's one region that's near the end, but not exactly at the end. This region at the end is required for pairing. I say here it's required in cis for pairing, and what that means is that it's required on this exact chromosome. This region at the end of the chromosome is required for the pairing of that chromosome. So what does that mean? It means, for instance, if you use some gamma rays and you break the end of the X chromosome and remove that pairing center from the X chromosome, now it can no longer find its partner. So even though both X chromosomes still have they have complete homology, they're identical DNA sequence along the rest of their length, they're missing this little region at one end. They're missing that region, so that makes them unable to pair. Now they still have chromosome ends, just like we do. So if we lost the end of our X chromosome, it wouldn't matter; they would still be able to pair. But in C. elegans, this white region on the X chromosome is extremely important. The reason why I'm showing you here that this white region has moved up to chromosome 1 is that this region also contains some essential genes. So if you completely lose it, then the worm will die. So you can't just chop it off. You have to put it on another chromosome. So not only do you need you don't need to have that on just one chromosome. You need, even if you have one of these regions on one chromosome, that's not enough. So each chromosome needs its own region in order to pair. That's kind of a complicated thing to say. If a chromosome is missing its pairing center, even if the other chromosome, even if its homologue has its pairing center, it will not be able to pair. So in this situation, there's also very little synapsis. So what are these pairing centers doing? Well, it turns out that each pairing center can bind to a certain protein, or a certain protein will sit on and bind to each pairing center. The X chromosome has a protein that binds to its pairing center, and this protein is called HIM8. And the autosomes 1 through 5 in C. elegans, it's a little bit interesting. So chromosomes 1 and 4 have the same protein binding to their pairing centers, which is called ZIM3. Chromosome 5 has a different protein binding to its pairing center called ZIM2, and then chromosomes 2 and 3 have again the same protein binding to their pairing centers called ZIM1. All these 4 proteins are very similar. They look very similar to each other, and the genes are very similar, and they're actually all these genes are very close together on the same chromosome. But they're all, each gene is also slightly different, and this ZIM1 protein only binds to chromosome 2 and 3, and it doesn't bind to the other chromosomes. Okay, so that's what the pairing centers are doing. They're recruiting, they're acting as a place for these ZIM or HIM proteins to bind to. And we know that's true because we can leave this pairing center intact on the X chromosome, but just delete this gene. We can mutate this HIM8 gene so that there is no HIM8 protein. There's no HIM8 bound to this pairing center anymore, right? So you usually have this HIM8 protein sitting on the X chromosome, if we remove that gene, now there's no more him8, and then again, the X chromosome cannot pair. So these pairing centers seem to simply be there to recruit these proteins. So there's a certain protein that binds to the X chromosome, and it's required for those X chromosomes to be able to pair with each other. And the same is true if you knock out the other one. So if you remove SIM2 protein, then chromosome 5 will not pair. But all the other chromosomes still pair fine. Okay, so what do these pairing center proteins do? Well, it turns out that they actually do just the same thing that the telomeres do in human cells. So it really is not anything specific. It's just these proteins come to the nuclear envelope and bring chromosomes also to the nuclear envelope. Although the difference is that they're only bringing one end of each chromosome to the nuclear envelope. So for instance, if you are missing the him8 protein, then the X chromosomes will not be at the nuclear envelope, but all the other chromosomes will be. And that means that over time, all the autosomes will be able to pair with each other, and they'll be able to recombine, but the X chromosomes will not. So you will not get any recombination between the X chromosomes, so the X chromosomes will segregate randomly. But that's okay in worms because just like in people, the sex chromosomes do not have to always be, you don't have to always have two of them. You can survive having a smaller number of X chromosomes. In fact, in C. elegans, XX animals are hermaphrodites. But if you're missing an X chromosome, then you are a male animal. So it's okay if you lose one X chromosome, you will still be a male. So animals that cannot pair their X chromosomes still survive. So it's a really nice system to study viruses. Okay, so after these chromosome ends are brought to the nuclear envelope, then what happens? There are a lot of other proteins sitting at the nuclear envelope waiting for them. I mentioned these last time, and these are the exact same. They're similar proteins that work in humans and yeast and plants. These pairing center proteins bind to an inner nuclear envelope protein called SUN1. SUN1 has some parts on the nucleus side, and then some parts sitting in between the inner and outer nuclear envelopes, inner and outer nuclear membranes. So the part that's sitting in between is able to interact with a different protein called Zyg12. This Zyg12 protein also bridges the outer nuclear membrane. It has some parts sitting in the space and some parts sitting outside. So between these three proteins, one completely in the nucleus, one that's in the inner membrane, one that's in the outer membrane, the chromosome is connected all the way to the cytoplasm outside the nucleus. And there it encounters microtubules and the dynein molecular motor, and so these patches, these complexes of inner and outer nuclear envelope proteins are able to move around on these microtubules. And I said last time I would show you a movie of it, and here I hope it works. So, okay, good. Fine. All right. So what I'm showing you here are each of these round circles is a separate nucleus, and in fact, it's a two-dimensional slice through the nucleus when chromosomes are actively searching for each other. The white patches you see are this Zyg12 protein, which is the one that sits in the outer nuclear envelope. And it's fused to this GFP, which is a green fluorescent protein that allows us to see it. So Zyg12 by itself, of course, is invisible, but if we put this GFP protein on it, then we can use fluorescence microscopy to see it. And I hope you can see, this is about 1 minute of total time. It's sped up. They don't really move this fast in real life. It really takes some actual time for these things to move. But I hope you can see that these patches do not just stay in one place, but they're kind of moving all around. They're kind of going out of focus as well, because this is just a two-dimensional slice. Let's see. But, yeah. But here I've taken these nuclei and projected the entire three-dimensional images into one two-dimensional plane. So now you can see all the patches of Zyg 12, and each one is connected to the chromosome end going into the nucleus. The reason it's getting dimmer over time is because I have to, in order to take a three-dimensional image, I need to take many, many images at different focal depths. And the more images I'm taking, I am bleaching the GFP. I'm destroying the GFP molecules by imaging them. So it gets a bit darker over time. I hope you can see, I think this is a really good example of this nucleus right here because all these patches are in a kind of one region of the nuclear envelope. And they're really kind of moving around each other. And so you can really imagine it's not just this little spot that's moving, but the entire chromosome is attached to it. And they're kind of sorting each other and kind of testing, are you my partner, are you my partner, are you my partner? And the more they, eventually they will go through all the combinations and then end up with the correct partner. So I think you probably also recall from last time that in C. elegans, you have this temporal gradient. So you start in early meiosis in this part of the gonad, and as you travel down the gonad, these cells are at a later stage in meiosis. So by looking at one region, you can see what's happening in early, middle, and later cells. And for instance, here chromosomes are unpaired, and here they are paired. And in this movie, you can see actually all three regions. If you look carefully, you'll notice that in these regions, you do not see these patches yet. So there's a lot of nuclei here. These are nuclei before they've entered this pairing stage, and they don't have Zyg12 protein yet because they're still at the interphase or leptotene stage of meiosis where they're not trying to find their partners yet, they're still just condensing. You don't see the DNA because we've only labeled this single protein with GFP, but of course the DNA is in there and the chromosomes are condensing. So from this part of the gonad, actually maybe from here, these nuclei have entered the zygotene stage and they're starting to pair with each other and they're starting to synapse. When they have finished synapsing, when they're completely finished, the Zyg12 disappears from all the chromosomes except one. And can anyone guess which chromosome does not it stays at? It's just the unique chromosome. The X chromosome, for some reason, keeps its patch of Zyg12, but all the other autosomes lose their Zyg12. So when you go down and see only a single patch, then you know that pairing is finished. So it only takes this length of time. A nucleus will travel from here down to here over the course of about 2 hours. So it takes about 2 hours for this pairing to finish. And you can see that it doesn't happen in all nuclei in exactly the same time. For instance, this nucleus seems to be finished. In fact, even this nucleus seems to be finished already. So it entered this zone of pairing up here, and by the time it got down here, it finished already. Whereas if you look at this nucleus and this nucleus, they're still trying. They're still trying to find their partners. So that kind of reflects the randomness of this process. Each nucleus, they don't have, chromosome 1 move toward each other. They don't know where they are to begin with. So depending on the state of the nucleus before pairing started and just how the chromosomes happen to randomly move into each other, that determines how fast or how quick these chromosomes finish pairing. Okay. So I showed you just the movement of all the Zyg12. This movie is a little bit different. This is just showing you the movement of the HIM8 protein, which, as you remember, is bound to the X chromosome. And also, all the chromosomes now are labeled because histone proteins DNA wraps around. Those are labeled with RFP, which is a red fluorescent protein. So this might give you some idea of how much the DNA itself is moving. Although there's so much DNA that it's kind of difficult to tell how fast these chromosomes are moving. But maybe this one you can tell chromosomes are really kind of dancing around and moving a lot. But if you look at the end, it's really moving a lot, right? The X chromosome, for instance, it makes a lot of these jumps. So each time the X chromosome jumps, you can think of it as it's grabbed onto a microtubule and moved along it as far as it can go, and then stops. And I'll give a little close-up of that. I'll just loop over and over again. I need to catch it at a certain spot. Okay. Let's see. Okay, let's move back. So this patch is the end of the X chromosome. And I hope you can see something that happens, something pretty drastic happens right about here. There. Okay. So what's happened here is that part of the X chromosome got on this microtubule and moved, but the rest of it was still sitting there. So what's happened at this point is that the end of the X chromosome really got stretched out. And this stretching of the X chromosome right here is thought to resemble the stretching in kinetochores that happens during mitosis, and maybe doing something very similar. It may be sending a signal that yes, this chromosome is finished pairing, because if this end of the chromosome gets stretched, then it can tell the cell that this chromosome is finished and we're ready to proceed. There's a very strong similarity between what's happening here and what's happening to kinetochores during mitosis. Okay, so you may think now, okay, so each chromosome has its own specific protein. So each chromosome has its own specific pairing center, and if they're missing that pairing center, that chromosome doesn't pair. And there's a certain protein that binds to each pairing center. So does that explain how chromosomes find each other? But in C. elegans, still the answer is no. We do not. We still don't know exactly what these proteins are doing. It seems like really all they're doing is bringing the chromosomes to the nuclear envelope. And since the same protein binds to two different chromosomes, we can tell right away that does not suffice. That doesn't give enough information to tell the difference, for instance, between chromosome 2 and chromosome 3. So even though chromosome 2 and 3 have the same protein bound to their pairing center, they never make a mistake and bind to the wrong, or pair with the wrong chromosome. So what started as something promising, because we thought originally maybe each protein or each chromosome has its own unique protein, and that is how the chromosomes find each other. It turned out that was not the case, and actually something else is also required for pairing. So just by finding these proteins and seeing how they work, that wasn't enough to understand how chromosomes pair. So to approach that question in C. elegans, one of the best and most powerful ways to study a process, any process, but in this case I'm talking about meiosis, is to ask a slightly different question. So we want to know how chromosomes pair, how they discriminate their partners. And this really elegant seeming explanation of each chromosome has its own protein, that turned out not to be true. So that was kind of, it's not a dead end, but that wasn't the easy answer. So we have to look for a more complicated answer. And so to do that, we can ask a different question, which is what genes are required for chromosome pairing? So instead of trying to figure out from first principles how might chromosomes pair, what we can do is we can mutate genes. We can break proteins and look at what happens and try to understand what proteins are involved in this process. And it's really easy to do that in C. elegans because if everything goes well, we can just look at the end product of meiosis and see that there should be 6 chromosomes. There should be 6 chromosomes at the end. But if one of these processes fails, so if pairing or synapsis fails, then at the end of meiosis, you may see more than 6 chromosomes. In this case, there is actually complete failure of meiotic pairing, and you will get 12 chromosomes. And that works for any stage, for anything that has to happen during this process. So if pairing fails or if recombination fails, then, as I said before, chromosomes will not be able to stay together after synapsis is removed. So even if chromosomes pair correctly, a failure in recombination will also result in more than 6 chromosomes at the end. So for the last little bit, I will describe one example of a gene, of a protein that's required for meiotic pairing, and that is a protein called PPH 4.1. That's kind of a long gene name, but we can just call it PP4 for short. It's called PP4 in humans, but C. elegans has some strange nomenclature for calling its genes. And so this is a mutant. This is a mutant allele of this gene, just like the mutant allele of the yellow gene gave you green color. A mutant allele of this PPH4 gene gives you bad meiosis, right? Gives you improper meiosis. So first I have to tell you what a phosphatase is, and to do that I have to tell you what protein phosphorylation is. Some of you may be familiar with this already, but it turns out that just having 20,000 different proteins in a cell, that may seem complicated enough to make a cell work, but actually proteins, once they're made, they're often changed. They're often modified chemically in order to change their function. And we've talked about one example of that already with the modification of histones. Histones can get methylated or phosphorylated to change their function. So we're saying this is an example of one of any protein. It could be, let's say this is a protein that's involved in promoting meiosis. This protein may be active, so the active form of this protein may require that it gets phosphorylated, meaning this PO4 group is put on this protein. A protein called a kinase is used to put these chemical groups on this protein, and in many cases, as a result of putting these phosphate groups on a protein, that increases the negative charge because the phosphate group has 3 negative charges on it. So that is often enough to change the shape of a protein, and changing the shape of a protein can often change its activity. Okay, so you can have a protein that requires phosphorylation to become active. So let's say this protein is active and it's doing something. Now this PP4 protein, PPH 4.1, comes along, and it's a phosphatase. And so the job of a phosphatase is actually to find those phosphate groups and take them off. Okay, so that's what the protein normally does. And when it does that, more often than not, that deactivates the protein. So you can think of PPH4 as a protein whose job it is to deactivate other proteins. In some rare cases, removing these groups actually activates the protein instead. So it's more correct to say that PPH4 is kind of like a switch that can turn other proteins on or off. So what happens if we mutate this gene? Well, we can change this gene in many ways. We can, of course, completely remove it, and then we don't have any activity at all. Or we can break it so that the part of the protein that normally is involved in removing these phosphate groups doesn't work anymore. So then, even if it hits this protein, it cannot remove those phosphate groups. So the protein stays active. So in a mutant, so if you have this gene mutated, this protein and many other proteins can become hyperactive, and so you get hyperactivation of certain processes. And in C. elegans, it's very easy to identify genes like this, because what happens is you just. So I said earlier that C. elegans are hermaphrodites, And if you take a single hermaphrodite, which has both male and female reproductive cells, if you put a single hermaphrodite worm on a plate, then in a few days you will have hundreds and hundreds of individual worms on this plate. And that's what they look like. Whereas if you have a mutant, if you have a gene like PPH4 that messes up meiosis, you will have very few worms on that plate later. And so just by taking a plate and looking at it, does it have a lot of worms or does it have very few worms, you can identify genes that give you meiotic phenotypes. Okay, so what happens in PPH4? If we have this mutated form of PPH4, I showed you these pictures earlier, but there's a little bit of nuance here that's very interesting. So if we count the number of chromosomes that we get at the end of meiosis, and each count we'll add to this bar graph that goes above the number we counted. So for instance, every time in the wild type we count 6 chromosomes, with this bar gets a little bit longer. So we can see in wild type animals, mostly meiosis succeeds 100% of the time. In fact, it always succeeds 100% of the time. This rare bit of 5 is just when 2 chromosomes happen to lay on top of each other. We still are honest and we count that as 5, but we know really, okay, that's 6 chromosomes. So in wild type worms, we always see 6 chromosomes. Okay, but in PPH4 mutant worms, we see a spread of different numbers of chromosomes. Almost never, so PPH4 worms, mutant worms, never really have all chromosomes properly paired. At least one chromosome pair is usually unpaired, and very often all 6 chromosomes fail to pair. So in this case where we see 12 chromosomes, 12 of these chromosomes at the end of meiosis, that means that all 6 homologous pairs of chromosomes fail to find their partner, and that's why we see 12. In some cases, one of the chromosomes paired normally. In some cases, 2 of the chromosomes paired normally. But more often, but always, some of the chromosomes fail to pair. So what's interesting about this PP4 mutant, this phosphatase mutant, is that it actually uncovers maternal age effect in these worms that's very similar to the maternal age effect that I told you about in humans. So in human females, when the age of the oocytes gets above 40 years, there's an increase in the number of aneuploidies and in the number of chromosome segregation errors. And so in these PPH4 mutants, you see the very same thing. If you look at wild-type worms, it doesn't matter if they're young or old, they never get worse at meiosis. In fact, so an old worm is actually only 3 days old. When worms reach 3 days old, they're about at the end of their reproductive lifespan. But even there at the end, worms are making 100% chromosome pairing. So they normally do not get worse at meiosis as they age. But if you are missing this PPH4 protein, so if you cannot deactivate these other proteins, then meiosis gets worse, and the number of complete failures of pairing, so the cases where you see 12 chromosomes, gets much higher. In fact, it gets about twice as bad. So overall, meiosis gets 2 or 3 times less efficient with just 3 days of aging if you are missing this PPH4 protein.
Excuse me, sorry, but just it's maybe not related, but if there are like cells with 5 pairs of chromosomes, why are there no cells with 7 pairs of chromosomes? Like, for example, in the first case, you have 5, but you don't have 7. And I mean, you have 7, but you don't have 5 for the PPH4 mutation.
Yeah, okay. Yeah, that's a good question. So the explanation is, the most likely explanation is that these cases of 5 chromosomes here are actually cases of 6 chromosomes. So they're actually cases where all 6 pairs of chromosomes did pair normally, but just in the microscopy image we have, 2 of those pairs are just sitting on each other. So we will still count that as 5. Whereas in this case, if we see 7, if you ever see 7, then you know that there's been a failure. Yeah, so 6 or less is most likely meiosis was perfect. But greater than 6 means there was a problem. Okay, so we know that all we know by looking at the end product. This is the diprotein diakinesis stage of meiosis when everything is finished, and we just look at the end result of how good pairing was. We can see that PPH4 mutation makes it worse, but that doesn't really tell us what went wrong because any of these processes could have gone wrong. So chromosomes pairing, getting close to each other, that might have failed. Chromosome synapsis could have failed, and then different steps on the way toward getting crossing over could also have failed. And failures in any of those stages could result in problems. In the last bit, I will focus just on the first two and see which of these processes might have gone wrong to understand where the problem is. I'm going to show you this again. Just to remind you that meiosis proceeds in space and time in this gonad. And what that means is we can look at the gonad and divide it up into equal-sized sections. We can call these zones 1 through 5, and then we can ask in each section what is the percentage of chromosomes that have paired and what's the percentage that are not paired. So by doing this, we can turn these pictures. We can turn these pictures of single or double signals into some quantitative data. So instead of having to show people pictures, here's a better view of this. So here's a signal on 2 different chromosomes that is visible as just one signal. That means the chromosomes have paired, whereas here, those chromosomes are still separate. Okay? So with these pictures, it's pretty easy to tell there's a problem with pairing over here, there's no problem with pairing over here. But by dividing the gonad up into zones and counting the total number of nuclei in each zone and the percentage that are paired and unpaired, we can go from these pictures to some quantitative data. And it's a little bit complicated here, but the main difference I can show you between the green and blue bars that are wild type levels of pairing and the yellow and red bars down here that are the mutant levels of pairing. So what you can see is that in wild type, before you enter meiosis, of course, there is no pairing, and then gradually the pairing levels increase, and this is pretty typical. They don't always go to 100% because sometimes chromosomes are still apart, but there's a very clear increase towards plateaus around 100% over time. Whereas in these mutant worms, the levels of pairing never go above 25%. Okay. So clearly there's a problem with chromosomes even just coming together and pairing with each other. One interesting wrinkle here is that the X chromosome doesn't seem to have a problem. The X chromosome uniquely can somehow, even without this PPH4 protein, the X chromosome can still find its partner. So pairing seems to have a problem. And then the next question is, if pairing has not worked normally, what about the next step of synapsis? What about the synaptonemal complex, the zipper that is zipping chromosomes together? So if you look at this and you compare normal, healthy, wild-type worms that can do meiosis perfectly with these mutant worms, it's a little complicated. So strangely enough, synapsis looks totally normal. So you can get synaptonemal complex, both the center zipper part as well as the axis part that's involved in condensation. Both of those elements are completely normally localized to all the chromosomes in both wild type and mutant worms. So losing this phosphatase protein does not prevent synapsis, so chromosomes still zip together just normally. However, there seems to be, that's kind of a contradiction. So I just told you chromosomes are not paired with each other. They're not paired homologously, but there's still synaptonemal complex connecting them. Just to take a closer look, so you can see the structure of the synaptonemal complex is still normal because the axis is in this dual kind of train track structure where the the axis that's, there's one axis for each chromosome. Those are on like the left and right, and then the central element is in between them. So anyway, the structure of the synaptic complex is the same, but if we take a closer look and do some computational tracing, we can see the difference between them. So here, these are wild-type meiotic nuclei, where all these chromosomes have been measured in 3 dimensions and stretched out. So the total length of the chromosomes can be seen here, and there's 6 chromosomes, as there should be, because in wild-type worms, chromosomes 1 through 5 and the X chromosome are paired and synapsed homologously. However, in PPH4 mutants, even though there is synaptic complex everywhere, we see more than 6 chromosomes. Okay, so this then can explain this apparent contradiction. Why do we see synaptic complex everywhere? So all chromosomes are zipped up, but they're not zipped up with the right chromosome. So instead of chromosome 5, let's say this is wild-type chromosome 5, they're normally paired with each other, and if you look at a region of chromosome 5 with fluorescence, they'll be merged together as a single spot. And then the synaptic complex will be between them. So this is what we would see in wild type. What's going on in the PPH4 mutant is more like this. So the chromosomes seem to be bent in half, and then these signals can be apart from each other, but they still have this synaptic complex zipping them off. Okay. So chromosomes are not paired, but they still get this synaptic complex between them. And so that's a really big difference, actually, between the wild type and this mutant, is that in wild type chromosomes, the synaptic complex is only between DNA that is homologous, DNA that could recombine. Whereas here, since it's the same chromosome, this DNA here is going to be very different from this DNA on that side. So even though the synaptic complex, these proteins are there, they're not in the right place. So both the pairing function and the synapsis function both seem to be messed up in this PPH4 mutant. And you can get very levels of chromosomes. Some have been folded in half. Sometimes you have 3 or more chromosomes all paired together. So you can have multiple chromosomes trying to all synapse with each other. So everywhere you have chromosomes, you have some synaptic complex, but it is not in the correct place. So chromosomes are not homologous. This is called non-homologous synapsis, and that's another thing that contributes to these problems. Okay, it seems like I'm kind of running out of time, so I'll have to get to the next part later. But the next thing we'll be talking a little bit about how we'll be going into some detail how recombination and crossover formation happens. And just to give it away, yes, these processes are also affected in this PPH4 mutant. So we'll be looking at how things work in wild type, normal situation, and also how they're messed up in this mutant. Okay, so that's it. That's all for today, and next week I'll have the quizzes graded and returned to you as well. Any questions?
The previous slides going to be loading on Panda?
Yeah, so I just figured it out today. I sent a mail, and I don't know if you will have access. There may still be some permission. Did anybody try it yet?
I accessed Panda, but
yeah.
No, this code does not appear in Panda.
Okay. Did you get the mail to Alexis? Did you receive a mail?
Yes.
And you click the link?
Click?
So yeah, you should have some mail link. Okay, that's not good. Yeah, I just started making this today, but yeah.
This URL.
Yeah, so if can I send this to you? Yeah, if you have that URL, maybe it will open for you now. I guess that's the next step, is if you can access this URL or not. Yeah, this link.
HTTP status 403. Access to the specified resource has been forbidden.
Forbidden. Okay, let's see. Maybe I can do. Maybe I need to do this. Yeah, it's possible that the permission is wrong then. So, yeah, I'm still kind of figuring this out.
Maybe some course registration, may be needed.
Okay, well, yeah, I will ask there's people I can ask to help me. So, okay, sorry.
Thank you.
Yeah, in the meantime, anyway, it's all there, so hopefully tomorrow I'll try to get this figured out.